So table 1 is showing screening guidelines. So the mean age was 31. The body mass index had to be overweight or obese patients. And then the 3-factor eating questionnaire measured cognitive restraint of eating, disinhibition, which is a tendency to overeat in the presence of stimuli such as food, and then hunger. So each of the 3-factor eating questionnaires, you had to have a minimal to moderate score in order to be in the study. So table 2 is showing comparison between the oxytocin and the placebo groups. So 72 hours prior to the study visit, this is showing food intake, so consumption of calories, carbohydrates, fats, and proteins. They were similar in both groups. A p-value was above 0.05, so in both the oxytocin and placebo groups. Sleep duration the previous night was also similar in both groups. Appetite as well as anxiety, so appetite. So they measured hunger and desire to eat favorite food both prior to drug administration of oxytocin and placebo, as well as 30 and 90 minutes following the drug administration. So they found that they were all similar in both groups. And they also found similar results in anxiety. So both prior and 30 minutes and 90 minutes following, they were both similar as well. So here we're measuring how oxytocin affects different anatomical areas of the brain. Brodmann's area, MNI coordinates are just 3-dimensional views of where these anatomical areas are located. So cluster size and voxels. So voxel is just resolution, so the bigger the cluster size, the better the resolution, which is ability to distinguish one structure from another. You have p-values, and then you have effect size, so level of activation. Here, oxytocin, there's greater contrast of oxytocin. So oxytocin leads to hyperactivation of the superior frontal gyrus and then the anterior cingulate cortex. And these regions are involved in cognitive control. So they're upregulated with oxytocin. And then here we have oxytocin leading to hypoactivation within these regions, which are mainly related to reward food motivation pathways. For zooming in on here, so the hypothalamus is important because it's essential for homeostatic control of feeding, and this is also downregulated in the presence of oxytocin. So here is a lateral view of the brain, superior view, and a posterior view. This image is basically showing the ventral tegmental area. So that's primarily responsible for dopamine pathway, the reward center in the brain, and they're just showing that's located in both the left hemisphere and then the right hemisphere. Here in the oxytocin group, in response to high-calorie food, they have a decrease in activation in the left hemisphere, and that's significantly greater than the placebo group. Here, in just looking at normal objects, there's no significant differences between the oxytocin and the placebo. And this is showing the right hemisphere. So they're just showing that the left hemisphere and the right hemisphere, oxytocin has a similar effect on decreasing the reward pathway within the VTA of the brain.